Or no? You set it to record? Okay. Sorry about that.
Ready? Okay. Well, good afternoon, everybody. Thank you for coming to this seminar. It is my pleasure to introduce Dr. Burt Bluhm. He is currently a professor at the Department of Entomology and Plant Pathology at the University of Arkansas. He got his master's and PhD degrees here at Purdue, actually at the Department of Botany and Plant Pathology. And I learned today that he also got his postdoc here with Dr. Larry Duncan. By the way, master's and PhD with Dr. Walshot. Dr. Bluhm has a background in pathology, mycology, and he also has expertise in molecular plant microbe interaction, functional genomics, metabolomics, particularly in the genus of Cercospora. His work has been cited almost 5,000 times, and today he's going to be talking about circadian clocks and cryptic hybridizations among Cercospora species. With that, let's welcome Dr. Bluhm.
Thank you so much for the introduction and the invitation to come back to Purdue. So it's been great. You know, it's like coming out of the pandemic, being actually able to see people in person and give a seminar, and to get lost on campus because they've rerouted all the roads since I was here. It's been wonderful. But also visiting with the students at lunch and old friends and some new faces. It's been really nice to be here. So today I'm going to talk about two distinct areas of our research. Some of our work in Cercospora has taken us to, within photobiology, has taken us to the circadian clock. And in soybean pathogens, Cercospora pathogens seem to be undergoing some very weird population level dynamics. So just a quick overview of the program. We work on a lot of different things, and Cercospora is definitely a main focus, but we, after the experience with Charlie, as a PhD student, I'll probably always still work on mycotoxins at some level. We work on a variety of soybean diseases, primarily Cercospora, but also Phomopsis, seed decay, a few other issues, a bit in sorghum as well. So Arkansas is on and off. It depends on the given season, but in some years we have over 500,000 acres of sorghum in the state. Works on in vegetable and fruit diseases out in California, so the table grapes and spinach. Some of this is with Jim Carell, that some of you may know, who's at the University of Arkansas. And then also we have a few projects on strange things, like we have a biological control project that I talked with a few people about earlier today, where we have a pigweed pathogen that is, it looks like a host-specific necrotizing toxin that we hope to eventually engineer as a bioherbicide. So into Cercospora. This is a slide that shows some examples of Cercospora diseases, and there is essentially a Cercospora disease that affects I would say every plant. We don't know that for a fact, but they're so prevalent and common that Cercospora is one of those genera that there's a Cercospora that's at least a nuisance, if not a major pathogen, of a wide variety of host species. Some are very important in tobacco. Cercospora ecosiana is very important, in soybean for sure. We also see a lot of unnamed Cercospora diseases on native and invasive plant species such as pigweed, and it's also very important on the rice. So just as a little more background on Cercospora for people that aren't familiar, one of the major components of Cercospora pathology is the production of Cercospring. So this is a polyketide-based photosensitizing proline quinone. Its biosynthesis is induced by light, and it is activated by light as well. So if you grow a Cercospora culture in constant light, you see this very distinctive red pigment. In darkness, you don't see it. It forms as crystals sometimes in the medium. And in the presence of light, Cercosprin enters an excited state that creates reactive oxygen species that result in cell death. So one important thing to consider is the the underlying gene cluster for Cercospring has been pretty well elucidated. This has changed a few times over the past 20 years, where the gene cluster, core cluster was discovered some 20 years ago and has been expanded over the last 20 years. And two genes to pay attention to that we'll come back to later. CTB1 encodes the polyketide synthase that is actually, excuse me, directly responsible for the production of the metabolite. And CFP is a gene involved in self-protection, export of Cercospora. So with gray leaf spot of maize, just a quick overview. We have, so there's two fungi that can cause gray leaf spot. Cercospora zeomatus is what we think of primarily in the US. It was the first fungus associated with the disease. In the older literature, you'll see this group 1, group 2 designation, it's group 1. Although there's no known sexual stage, it has a predicted Mycosporella telomorph. And Cercospora zeana is another related Cercospora that causes the exact same disease. It's been described as a distinct species. Looks very similar in culture, but there are some subtle differences. It doesn't grow as a rectangle. You can actually pack conidia out to get it to coordinate conidiation. We'll be talking mainly today about Cercospora zeomatus. So management of gray leaf spot or GLS is very problematic. In the US, we think of this as kind of a production cost sort of issue. So the input costs that are required to manage the disease can be considerable. In other regions of the world, where especially smallholder farmers, where there's not a lot of tools available, this can be a very devastating disease, where especially if, like in areas of Africa where you have open pollinated varieties, there's no good genetic resistance, there's no good mechanisms of control. And so you can see, like in the US, we think about, humid conditions. We can, in a typical year, you may see this level of incidence and severity. Under pivot irrigation can be much higher because of favorable conditions. But in other parts of the world, like these pictures were taken in China, these are fields that are absolutely devastated by GLS. And you can see where even mummification of the ears itself in very, very severe cases, and certainly correspondingly yield loss. So I'll start this story a little bit with, we're kind of picking up where when I was a postdoc here with Larry Dunkel, Larry was very interested in photobiology, and Cercospora is a good system for that. And so at that time, this was around 2007 or 8 when we initiated the project. It's important to note this is kind of before the real advent of next-gen sequencing. The tools and resources that were available were fairly limited compared to what they are now. And so we were able to, in the case of Cercospora, there were no genomes available, a handful of EST collections. This was kind of a much more difficult process. So we were able to fish out an ortholog of the Neurospora white collar 1 gene, and we named that CRP1. So in Neurospora, the white collar complex governs a lot of responses to light, including the circadian clock, secondary metabolism, conidiation, a few other traits. This ortholog that we were able to pull out, we compared it at the time. So this is structurally, as far as like conserved domain structure and other things, it looks, that wouldn't be going on. Yeah. It looks very much like the white collar one. So we, at that time, based on predictive analyses, we were fairly confident and modeled the love domains, the light, oxygen, voltage domains. And so we knocked this out through insertional mutagenesis. So we weren't able at the time, we didn't have enough sequence information to actually fully delete the gene, but we were able to insertionally disrupt it and then characterize that mutant. And so what we found was that in the CRP1 deletion strains, we did not see the onset of lesion formation or mature lesions to form. And when we looked at this in a little bit more depth, part of the issue was that the CRP1 mutants seemed to be unable to sense stomata. So in Cercospora zeomatus, after spores land on the leaf and germinate, those germ tubes are able to sense stomata and then form an aprosorium and infect through stomata. And what we saw in the CRP1 mutant is they were essentially blind to stomatas when it looked to us. So we saw phenomena where they would grow across indiscriminately or even just grow around. And so you can see here, like this is with GFP labeling subsequently. And per stomate encountered, the percent of Apresorium formation, it wasn't absolutely zero, but it was very low. And whatever Apresoria formed appeared to be inviolable. So we started there with the question, and this was about the time that I was moving to Arkansas, with this kind of basic question. So this is just a very kind of rough framework, conceptual model of trying to formulate hypotheses as to how Cercospora zeomatus used blue light and the circadian clock potentially to regulate pathogenesis. And in this working model, you can see there's a lot more question marks and black boxes at that time than there were details. So just a quick overview of the fungal circadian clock. So the white collar complex is a heterodimer. There's white collar 1 and white collar 2, which in our system corresponds to CRP1 and CRP2. Their activity is induced by light, which then in turn activates the negative feedback loop of the clock, which is predominantly mediated by a gene called frequency or frequency. And so frequency is induced by light, its expression is induced by light, and the protein is eventually turned over. When the level of protein dips below a certain level, the cycle repeats. And so one important thing, and we may mention this again later, so to be truly circadian, the oscillation, the cycle, has to be independent of temperature and other factors, has to be entrainable, in this case by light, and has to be free running in the absence of external cues. So basically, if you were to entrain a fungus and activated circadian clock, if you put it in darkness, that cycle has to still run on a roughly 24-hour period. So one of my graduate students that some of you know, John Ridenauer, worked on this project for his PhD. And one of the first things he did is he went back shortly after leaving Arkansas with the help of Steve Goodwin, we were able to get a much more finished genome, much more complete genome through collaboration with DOE JGI. And with that information and the acceleration of improved techniques for functional genomics. So one of the first things John did was to go ahead and knock the gene out completely delete that open reading frame. And he confirmed that basically in that case, so certainly that complex regulates, the CRP complex regulates kinetiation. So in constant darkness, you see with the wild type that you have a high level of kinetiation, 2 independent mutants, very low level, complemented strain is restored in a light-dark cycle. The wild type, that's sufficient light to suppress kinetiation. And intriguingly, the mutants then do produce kinetia in 12 light-dark cycles. And so looking at this, so germination is not affected. So what spores are produced in different conditions are viable, but the induction of kinetiation is very much controlled by this complex. And so very quickly, John also knocked out the CRP2 gene. And just to confirm or to try to dissect the role of different components of that heterodimer, and without going through this figure by figure, you can see in parallel very similar translomes. They're all essentially indistinguishable, where the CRP2 mutant behave the same way as the CRP1 mutant as far as those traits. So when it comes to regulating kinetiation, the 2 components of this heterodimer seem to be roughly equivalent. So also confirming that the CRP complex regulates pre-penetration infectious development with this full-length deletion, 2 independent strains, we saw no lesions formed. And to dive in a little bit deeper with the phenomenon of why that was the case, looking at the CRP1 mutant, it was able to encounter stomata as efficiently, to sense them probably as efficiently as the wild type. It was not able to form Apresoria at the same sort of rate, and that percentage was also correlated to the ability of the fungus to sense stomata. So for every stomate that it sensed, it still was not able correspondingly to produce Apresoria. And here it just shows CRP2, very similar to CRP1, where so again, like when it comes to that particular component sensing stomata, it looks like both components of that are dying or indistinguishable. So we move to look at frequency, and so the circadian clock in Neurospora has been studied extensively, and frequency is a key component. But Neurospora is not a plant pathogen, so investigating the role of some of these phenomena, especially frequency, is not really possible in that system. So we found an ortholog, convincing ortholog of frequency in Cercospora xenomatus. You see the conserved domains here, very, very highly conserved in our opinion. And then also through homologous recombination, deleted the entire open reading frame of frequency. And so what we saw was that frequency is induced by light, as you would expect for a clock component. That induction requires CRP1. So in 2 independent knockout strains of CRP1, we did not get that induction. And one of the most interesting things out of this was the oscillation of frequency expression in Cercospora xenomatus mirrored a typical circadian sort of oscillation. So this was really the first direct confirmation that Cercospora possesses a circadian clock. And that's kind of important because the number of circadian clocks that have been established in fungi is not that extensive. And exploring that role in plant pathology hasn't really been deeply explored as well. So an interesting finding with frequency is that frequency is definitely required for pathogenesis. You can see here the 2 independent deletion strains, no lesions formed after 14 days. And that the underlying dynamics there were interesting, that we saw a slight reduction in the ability of frequency possibly to sense stomata, and the number of aprosoria formed was as low or lower than in the case of the CRP1 and CRP2 deletion strains. So similar overall as far as the CRP1 mutants in these components of pathogenesis, but not quite identical. And perhaps the most interesting thing past that was so so the overexpression of frequency in the wild type did not substantially affect pathogenesis. So when we see the wild type here, the number of lesions induced, the overall level of necrotic colonization on the leaves, fairly similar when you overexpress frequency in the wild type strain. These are 2 different overexpression lines. But what was really interesting to us, particularly interesting, is that in the CRP1 deletion background, so this is the complementation strain right here, where we have virulence restored. If you overexpress frequency in a CRP1 deletion background, you do get the initiation and formation of lesions. It's not quite a full restoration of the wild type phenotype, but it's a substantial restoration. And so that would suggest if you have, if the role of CRP1 in that complex was entirely, if that was the primary governor of pathogenesis, to be able to overexpress frequency in that background suggests that frequency has some clock-independent function in the regulation of pathogenesis, which is an interesting finding. So both CRP1 and frequency are involved in the production of and resistance to Cercospora. So this is also a very interesting finding where if you delete CRP1, then you reduce the amount of Cercospora that's produced in light, but it's de-repressed in darkness. And in the frequency mutants, 2 independent strains, Cercospora biosynthesis is essentially blocked. If you overexpress frequency, it's irrespective of light or dark. And so working through how exactly Cercospora, which is induced by blue light through the CRP1 complex, the de-repression, that part is very interesting. And so Another interesting thing is that when you look at the ability of the CRP1 or frequency deletion strains to respond to Cercospora. So Cercospora as a metabolite is self-toxic. The fungus has to have an active mechanism to either export it, neutralize it, something has to happen because it's equally as toxic to fungi as it is to plants. And if you look here, you can see the exogenous application. These are just dilution plates of spores. So CR is Cercospring. If you add either 1 micromolar or 10 micromolar Cercospring, what you see is that the frequency mutants are considerably less able to protect themselves against exogenous Cercospring. So we see somewhat of a reduction, a slight reduction in CRP1 mutants here, as you can see, compared to the wild type control, but the frequency mutants are substantially impaired. And so this led us to kind of, untangling the role of the clock, the circadian clock and pathogenesis in the system is really challenging. So we didn't see a clear relationship between the clock and stomatal infection. We didn't see, other phenomena that we could, conidiation appears to be, controlled by light and the clock, but that's not, is directly relevant to pathogenesis. And so what we saw, we had a few discussions at, Journal Club and over coffee and over a beer about what could possibly be going on. And one of the ideas that we kicked around was that, what if Cercospring is actually under clock control? So that would make sense that, perhaps self-protection and or the production of Cercospring would be under clock control. And so we found that that's the case. So that's the clearest link that we found between the circadian clock and pathogenesis. So relative expression levels in constant darkness, this is in free running conditions. So back to that idea of train the clock and then do it in free running conditions. So frequency exhibits this is subjective darkness and subjective light. This is what the fungus would be remembering as light or darkness when it's in free running conditions. We see a circadian oscillation in frequency expression. We also see a similar oscillation in the expression of CFP1, which is one of the major genes involved in self-protection. So the fungus is basically, in our working hypothesis, using the clock to anticipate when Cercospring will be active, biologically active. So the exporter is induced immediately before dawn, and therefore when Cercospring is present and is activated by the light, that export function is at its highest. And we also saw another gene, CZK3, a MAP kinase that is also under clock control. And intriguingly, CTB1, which is the polyketide synthase, its expression peaked in, I guess, the hours before dawn, you could say. So in anticipation of daytime exposure to light, that polyketide synthase was expressed at the highest levels. And so that makes sense too. So the fungus is anticipating light, it's gearing up the production of the polyketide and Cercospring in the night before daylight. And as daylight approaches, we see the induction of the symporter that is involved in self-protection. So that was the closest link that we could see regarding the circadian clock and this mechanism of pathogenesis in Cercospora. So another interesting way to look at this is, kind of the final part of this first phase of the talk is, so in this experiment, we entrained the fungus as described. So we activated the circadian clock and then let it in free running conditions run. And if you pull the fungus out of free running conditions, either when the fungus is experiencing subjective day or subjective night, its ability to protect itself against Cercospora varies. And so no Cercospora control. So this is change in conductance. This is electrolyte leakage out of cells that we could monitor in subjective evening, then the fungus was much less prepared to protect itself against the exogenous application of Cercospora than it was in subjective morning. And so this is more of a physiological sort of confirmation to the phenomenon that we saw at the expression level. And it's an intriguing link between not only secondary metabolism, but a very important phytotoxin in the way that the fungus is carefully coordinating its efforts. I think this kind of gets overlooked sometimes, where we see how the average fungal genome has, who knows, between say 20 and 50 secondary metabolite clusters, and these are all having to be regulated at different levels, coordinately regulated, and presumably have fairly specific ecological functions. And how tightly those different metabolites are regulated biosynthetically, it makes sense that this would be, at least for some of them that are self-toxic, a very intricately regulated process. So I'll shift gears now to Cercospora leaf blight and purple seed stain. So this is these two diseases, and I'll mainly talk about it in terms of Cercospora leaf blight, but these are also important Cercospora diseases of soybean that we see very commonly in the South, and maybe not as commonly in the Midwest, but these are diseases that can lead to, in mild symptomology expression, bronzing, this kind of purpling and mottling, a lot of times necrotic leaves that ultimately, and I don't have a picture here, but it can almost look like the field was burned with the chemical application. So at maybe R2 stage or even R1, R3, the necrosis in the petioles causes the leaves to drop. And this is an extreme effect on yield when this sort of thing happens. These fungi can also colonize seeds and get this kind of purpling the seed, which is important for dockage. In 2018, there was concerns in the feed industry for the first time, feed refusal for animals that were being fed feed derived from soybean that had very high levels of purple seed stain. So whether that's Cercospora or not is another issue, but there's probably also other toxic secondary metabolites that very severe conditions of purple seed stain affect human animal health. So Cercospora associated with soybean, we historically thought of this as a species called Cercospora kikuchii, or some people say kikuchi. And for a long, long time, almost 100 years, it was believed that Cercospora kikuchii was the sole causal agent of purple seed stain and Cercospora leaf blight. In more recent years, Cercospora CF flagellaris, which is kind of this taxonomic distinction for an unfully described species, and Sisbeckia have also been associated with Cercospora leaf blight. And so there's a couple papers that have come out. We were co-author on the one PLOS one, and then another out of LSU, roughly at the same time, documenting the same phenomenon in the US and especially in South America, where it was unclear to what extent initially that Cercospora kikuchii might have been replaced, but certainly these two other species, apparently in a relatively short amount of time, ascended rapidly to become dominant. And so we started this part of the project by wondering, to take a snapshot to see, okay, like what are these dynamics in real time, or at least have a benchmark approximately 10 years ago in case this is changing quickly or dramatically. And then, of course, curious about the underlying mechanisms of this shift. So we obtained a large collection of pathogens of isolates from all kinds of hosts, primarily soybean, but lots of different hosts as well, other agronomic crops, weedy species. And some of these we were able to analyze through whole genome sequencing and resequencing. We developed, and this is a few years back, so sequencing has evolved very quickly. So kind of a hybrid sort of assembly approach for fairly complete genomes. But today, with PacBio, 50x, 100x coverage would probably be a better approach. And so we were looking at this and then evaluating host specificity so that we could move towards elucidating mechanisms. So we collected heavily out of the states of red and isolated from leaves. Some of the members of the audience or maybe on the Zoom have isolated Cercospora from leaves. It's a bit of an art in its own self, in its own right. So it's a process of getting these leaves to getting conidiation and picking individual conidia and then getting pure cultures. And so we, at the time, collected about 700 isolates. We're probably today closer to about 3,000 that we have in the collection from this project. And we identified it all based on morphology because Cercospora taxonomy is very distinct from Flagellaris and Sisbeckia. Barcoding for 168. At this time, this project is ongoing and whole genome resequencing for about 28. And what we found from that sampling over a couple of years, just immediately before the pandemic, was that Flagellaris represented 98% of what we had. Sisbeckia, maybe about 2%. It's a little harder morphologically to distinguish from Flagellaris. Our confidence isn't quite as high. Our confidence is very high with Capucci, and we saw 0. So it looks like complete replacement. And we've talked to collaborators in these states who also are performing their own isolations. And the original causal agent seems to have roughly vanished off the face of the map, as far as we can tell. And it's relatively quickly because our reference strains of Cercospora taxonomy was collected by Scott Abney in the early 90s here in Indiana. And so what we have a few isolates from Scott's collection that are labeled Cercospora taxonomy associated with purple seed stain and Cercospora leaf blight, and they're all true Cercospora taxonomy. And so sampling Indiana today, all Flagellaris. So it's relatively quick. When we look at diving into the level of genome sequence conservation, there's several pairwise comparisons here. So Viticula here, we see at the level of genome identity being perfect identity here, clonal, we see that a lot of these pairwise comparisons are fairly well conserved as you would expect for species within the same genus, but not incredibly well conserved. It's important to note that like Flagellaris and Capucci are still only at the maybe 0.92 sort of level. But if you look at the comparison between Cercospora sisbeckia and Cercospora taxonomy, the taxonomy that was collected in Indiana in the early 90s, the Sisbeckia that was collected in Louisiana in I think 2014. So this is that same scale as here, and the level of identity is so compressed that you have to zoom in on the axis to appreciate that. There's essentially almost clone. And when at the level of assembly that we had at the time, when you look at the alignment between, say, Flagellaris and Sagiana or Flagellaris and Capucci, the number of highly conserved regions is relatively low. If you look at Sisbeckia versus Capucci, it's essentially the whole genome. And so it's interesting to us because Sisbeckia morphologically and in the regulation of other things like Cercospring, for example, is clearly a distinct fungus compared to Capucci. You could hold the two plates up and tell very clearly that they're two distinct organisms. And so we were able, in this same sequencing and sampling approach, we found one isolate of Sisbeckia in Arkansas in 2017. So we resequenced the genome of that isolate, since Sisbeckia was relatively uncommon. I'll kind of run through Sisbeckia first and then go to Flagellaris. But yeah, so we resequenced that one isolate because we're curious. We didn't have as much information about Sisbeckia. And so if you compare the 2017 isolate to the 2014 reference isolate that was collected in Louisiana. This is, so this is basically, you can think of this as like a genome synteny map. A flat line means complete conservation and identity. We found that even with that sort of geographic distance and time, those two isolates were essentially cloned. There's a handful of SNPs scattered throughout the genome. When we compared Cercospora taxonomy collected by Scott Abney to the 2014 strain from Louisiana, we saw regions of very high identity, but we would then see these kind of pockets of much lower genomic identity. So it almost looks like an introgression block. And that's our working hypothesis, is that there was some other Cercospora that was able to form a hybrid with Cercospora taxonomy, and there's potentially one event or maybe a very low number of events. And that particular lineage is stable as an asexual clonal organism. But one thing that jumps out is, so Cercospora taxonomy has followed this sort of pattern where, you know, the barcoding approach, you have 4 loci to resolve species within a genus, 1 deviates, you find a 5th locus, and that doesn't work as well, you find a 6th, 7th, 8th, and so on. And in Cercospora, you know, this fundamentally works, right? So if you just kind of think about, okay, so say you have actin and EF1 alpha, ITS. So if these are, if evolution follows the route that you would expect, then you should be able to distinguish Cercospora taxonomy with these loci, because every other member of that group has evolved separately and in isolation. The sequences should work as barcoded loci. But what we've seen out of Pedro Krause's group and others is that these typical barcoding loci, it seems like there's always a deviating isolate. So you can have 7 barcoding loci and use those to resolve, say, 40 or 50 strains, and it works for most of them, but then 1 deviates. And that level of deviation is part of what drove the search for more and more barcoding loci and why that's expanded. But what we see in the example, a very specific example of the same alignment, this putative recombination plot, is we've seen some of the canonical barcoding loci within those recombination blocks. And so if that's the case, that would explain perfectly why they get these deviating isolates and why they keep needing to add more and more barcoding loci to try to resolve hybrids, which theoretically can never be fully resolved as long as hybridization is ongoing. And so just to illustrate that point, you can imagine that if you have some introgression or hybridization event where you have an unknown Cercospora species contributing the actin locus from its genome, and then perhaps in the other direction, you're simply not going to get very usable results with barcoding loci. And so if this is the case, it probably requires, and I think this is not unique to Cercospora, but it requires a rethinking maybe fundamentally of the species complex or concept, where, you know, if you have the propensity for this level of interspecific hybridization and recombination within the genome, that defies kind of the core species concept that's used for most fungal taxonomy. So in the case of Flagellaris, it's a little different. It's a little more complex. We had 28 isolates that we resequenced the entire genome, and using whole genome comparisons for phylogeny, then we found 4 distinct lineages so far. And these lineages, as you can see, these colors kind of roughly correspond, the blue, the green as well. When looking at the geographical distribution, there doesn't appear to be a geographical explanation to the distribution of these lineages. We saw all the lineages from essentially across the entire US. like Iowa, for example, 3 distinct lineages out of 3 genome sequenced in this case. And so similarly with whole genome alignments, these are all the different, it may be a little hard, unfortunately, to see the color there, but if these are the blue, the gray, the green, and the orange. So we can look across all of the isolates and see some very, very well conserved regions, and some regions that are much less conserved across all isolates. And you can see a little bit of that pattern here, for example. But what also points to inter and or intra-specific hybridization within this group is when we look at potential recombination blocks, we see some, like within this orange clade, that these, so I should mention that when you see the break in the line, that's a loss of synteny. So as you look across these aligned genomes, where you see straight lines is very high levels of identity conservation of synteny. And where you see breaks is regions where it's much less conserved. And the pattern in the break you can kind of see here is sometimes conserved as well. And so in this case, within this one lineage, we see what looks like potential introgression from another lineage. Those are conserved. But in other cases, we see in the same isolate where other blocks appear to be better conserved than others in the same scaffold. And so that would suggest that there are, if in the event of interspecific hybridization, that this has probably been ongoing multiple times. If it were a single hybridization event, we'd probably only expect to see one region or isolated regions holding a similar pattern. But when you see different patterns within and across different isolates, that suggests that it's gone on multiple times. And so we're really curious about who the parents are. Unfortunately, there's just not enough resources in Cercospora genomics to know. We have ruled out a few suspects. So Cercospora sagina, which causes frog eye leaf spot, does not appear to be the parent for these, or to have contributed meaningfully. A few other Cercospora we've also ruled out. So we don't know at this stage who the parent is or parents. But one thing that's also unusual about Flagellaris is within Cercospora, the original kind of species concepts were based on host association. And so Charles Chuck did this work throughout his career. He published a monograph in the 50s that very carefully outlines phyllide length, number, and size, number of conidia produced per conidia, average length of conidia. So based on kind of classical morphological characteristics. And there was good evidence at that time that there were distinct Cercospora species that were very host limited, maybe to a given host genus or even a host species. In the case of Flagellaris, it defies that. So we have found this Flagellaris group associated with highly diverse hosts, monocots, dicots, agricultural species, weedy species, native trees. There's Flagellaris can be found in, associated with many, many diverse hosts. And individual Cercospora flagellaris strains have an unexpectedly expanded host range. So the association of Cercospora flagellaris with diverse hosts itself does not necessarily prove that a given isolate is able to broadly infect. Those could all be distinct lineages that are just related enough to still be within this clade. But what we saw was that isolates from a flagellaris from Cercospora leaf blight can infect Palmer pigweed. So this is a very common noxious weed in soybean production throughout the U.S. or increasingly throughout the U.S. Isolates from soybean can infect different vegetable crops, and isolates from pigweed can cause purple seed stain in soybean. It's very difficult to get the expression of symptoms of CLB in greenhouse conditions, and so this has always been kind of a bit of of a frustration. There's detached leaf assays that work fairly well, but it's a detached leaf assay, so it has its own pros and cons. And so, but purple seed stain, we can fairly reliably get symptomology. And so the important point here is that individual isolates of the Cercospora flagellaris clade are defying this convention of being limited to a specific host. And this would also fit with the idea of, potentially if you have a group of interspecific hybrids who have shuffled their effector complements, perhaps this broader host association, there's enough novel combinations that they've achieved or broken free, I guess you could say, of host specificity. But one important point here, if you notice that the vegetable crops selected at the bottom are all brassicas. And so we're very curious in the case of brassicas if we could develop a Cercospora arabidopsis pathosystem. And we were successful. So the picture down here on the bottom left is from the Compendium of Brassica Diseases. So Cercospora symptomology on different hosts can be very different, from like a bronzing at CLB versus distinct frog eye type spots. There's a lot of frog eye Cercospora diseases to kind of these whitish, bleached out sort of lesions. And so among the brassicas, these whiter lesions are the most common, commonly associated with Cercospora. And so what we found was that multiple Cercospora flagellaris isolates from soybean can infect arabidopsis, as seen here, and flagellaris isolates from different hosts. So these are different here, and here are all from soybean. We were able to isolate flagellaris from a mustard crop in Arkansas. It also was able to infect arabidopsis, and a watermelon isolate was also able to infect arabidopsis. So this is we're hoping will be a system that's broadly useful, not only to the community, but as a mechanism from a very practical sense, to be able to screen potential R genes. So like via Higgs host-induced gene silencing, to create those transgenic lines in soybean and then to evaluate those would be a very costly and kind of time-consuming process, where we're hoping we can evaluate those much more quickly in a arabidopsis sort of system, as well as drill down into some of the basic concepts of host resistance. Because one thing that we've noticed just anecdotally is there's a lot of wild brassica weeds other than arabidopsis. And a lot of these, at least in the southern U.S., tend to be very early season weeds and sometimes late season weeds. And they're just disgustingly healthy. And we have never been able to find a Cercospora on these native weed populations yet, and extensively looking. Some of that could be the timing. These are, a lot of these in the south at least, come out very early in the season. So there hasn't been an inoculum from field conditions or other crops. Conditions aren't probably as favorable for Cercospora. We're a little bit surprised at some of the late season ones. We haven't seen it, but again, a lot of these weeds have a very short lifespan, and so perhaps they're avoiding it so we're not sure. Another possibility is that in some of the native plant systems, there is some level of endogenous host resistance. And so if we could find that, mine that out, then that would also be a potential value of the arabidopsis study system. So I'll shift over a little bit to talking about some mitochondrial genome dynamics among these two. There's a few things we've seen here that are curious that we're still pursuing and frankly don't fully understand. So we sequenced the nuclear genomes of Scott Abney's Kikuchii, the Sisbeckia from LSU, our flagellaris kind of type strain, and Cercospora saugina. And we're also able to assemble complete mitochondrial genomes. So this is just a quick comparison of that. But more importantly, what we see, we're exploring the hypothesis that there is mitochondrial introgression among some of these groups of Cercospora. So when we align mitochondrial genomes, for example, from Flagellaris, Kikuchii, Sisbeckia, and Cercospora reticola, which has been reported in New York and I think other states, that and sampled in the field, a substantial percent of the isolates actually can be Cercospora flagellaris. So this may be a case where, in the case of Cercospora reticola, we're seeing in real time this takeover of the historical Cercospora associated with the disease and this flagellaris group. Time will tell. That's just one idea. But it's interesting that of those 4 known species, or these 2 historical and these 2 more modern, there is an extremely high degree of conservation among mitochondrial genomes. It's a very condensed mitochondrial genome, very high level of conservation. And with Cercospora sagina, Cercospora zymatus, and there's a few other that we have now that aren't on this slide, we just don't see that level of conservation at all. So it's strange that there would be a maternal, potentially mitochondrial genome, and that might explain at some level, or it might at least be correlated with the mechanism underlying this potential hybridization within these groups. So just one other curious observation is that within Cercospora flagellaris, the mitochondrial genome was very highly conserved across isolates, but there were there were 3 relatively small indels that distinguished those mitochondrial sequences into 3 groups. And so if you look at this, there's a very small insertion here, right? It may show up. So it's group 2, circled in blue. Group 3 was a very small group and had another larger insertion there. Group 1 all shared the same sequences. So most of what we saw was grouped so far. And what was curious was, so we didn't see a clear association between genomic and mitochondrial haplotypes. So this shows here those same lineages that we talked about earlier within flagellaris. So the blue, the orange, the gray, and the green. As you can see, within this lineage, we saw mitochondrial haplotypes 2 and 3. Within this lineage, 1 and 2. Only haplotype 2 in these 2 genomic lineages, but a much smaller representation so far. We didn't see an association with geographical distribution. So haplotype 2 was found, for example, in many different states. We didn't see a clear association by host. So whether it was soybean or cotton, the potential group 2, or within soybean 1 or 2, or 3. And the G143a mutation that conveys resistance to strobilurin fungicides is present in most lineages, as you can see here by the A distinction. But one particularly interesting thing was that we found the G143a mutation in an isolate collected from watermelon grown in a home garden, substantial distance from row crop agriculture. So in the state of Arkansas, all the row crop agriculture predominantly is in the eastern half of the state. And this was collected from a home garden in the Ozarks, long distance from where you would expect there to have been any history of strobilurin application. So this seemed a bit out of place and was a clue that perhaps the geographical movement of these hybrids is more than what we might have guessed, potentially because of that broad host range. So we suspect that the members of this group are moving around quite a bit physically. So this has kind of taken us to a consideration about how to view pathogenesis in Cercospora flagellaris. We think that host specificity is more likely associated with the latent phase of infection In other Cercospora, we've seen that if you if you inoculate spores onto a non-host leaf, that the fungus is still able to germinate and find stomata and generally produce an amprosorium, and then that's where the interaction stops. And so probably specificity is more with that latent phase, which would be consistent with effector-driven manipulation of host defenses. And so if there are one or more or many Cercospora species that, for whatever reason, relatively recently have gained the ability to form inter and then intra-specific hybrids, this entire complement of effectors could be getting shuffled around in many, many different possible combinations. And so when a Cercospora has evolved in isolation of a given host to be able to effectively spend its evolutionary energy on developing an effective effector catalog, then the end run on evolution would be then to share that in some combination with other isolates that had done the same, thus broadening out or altering host range. So there are some practical implications, especially for resistance breeding in soybeans. So Cercospora leaf blight is a disease that there's very little genetic resistance. This disease has reemerged relatively recently in the last 20 or so years, and in Arkansas, maybe the last 10 to 12. It was one of those diseases that kind of felt like it came out of nowhere and was severe very quickly. And so breeders are, public and private sector breeders are making efforts to develop resistant cultivars. But if you have this degree of, even if this is a fixed level of hybridization, and it's never going to happen again, it appears that there's a sufficient number of hybrids out there that there's a lot to account for as far as breeding for resistance. And so knowing what to breed against is a problem for breeders. The broad host range from the point of view of practical management. So if you think about, like in the South, things like kudzu that stay green all winter, and Cercospora flagellaris can infect it. So as a potential source of inoculum, a green bridge to overwinter and survive, that's a practical consideration. And so, we've seen, at least anecdotally, where especially at first, it was very frustrating for breeders who would screen germplasm in distinct locations, and something that looked very resistant in Louisiana held up very poorly in Missouri. And so this disconnect seemed to be more driven, instead of environmentally, more likely was driven by pathogen diversity. And so the durability of resistance is also a question. We don't know how dynamic this process might be. We don't know how quickly the potential for new hybrids to emerge or to ascend at the population level might be. So a lot of unanswered questions on that front. And it kind of, raises the question conceptually, is it better to try to suppress infection or control the development of necrotic symptoms? Because the main problem that we see with CLB, at least in the South, is if it comes in early enough, or if you have late planted soybean, it can completely defoliate during grain fill. So if you could avoid that defoliation, that would be a huge plus. All right, so a few top take-home points. So in the case of Cercospora zeomatus, we see that it possesses a circadian clock, which is an important finding in and of itself. It's one of the very few fungi known demonstrated to possess a circadian clock. We knew that the CRP complex, was heavily involved in pathogenesis, but a more thorough evaluation helped establish roles in secondary metabolism, regulation of secondary metabolism, pathogenesis, condiviation, infectious development. Frequency of Cercospora zeomatus may play a clock-independent role in pathogenesis, which would be a very kind of interesting development. So far as that $1 million why does Cercospora zeomatus have a circadian clock, or how does it use it? The clearest link that we've seen so far is probably in the regulation of Cercospora biosynthesis, as well as self-protection against Cercospora. Those two seem to be fairly clear. In soybean, it looks like Kikuchi, we can't prove the negative, so we can't say that it's gone, but it seems to have been substantially diminished by the ascendance of Cercospora flagellaris and, to a lesser extent, Cercospora sysbeckia. And it's interesting that Cercospora sysbeckia is much closer to the original Cercospora kikuchi. So essentially flagellaris, if you were to go to a soybean field and pick a leaf showing CLB symptoms, it's most likely flagellaris in the US. As noted, sysbeckia, it seems to be very clonal and maybe even originating from a single hybridization event. It seems to be, we have very limited information on it, so we can't say conclusively, but so far we're in the process of sequencing more isolates to have a little bit more support for this. But it's very telling, with that level of physical separation and temporal separation, that two isolates were essentially clonal. Flagellaris is much more widespread. The genomic dynamics seem to be much more complicated. We were very surprised at the broad host range. It's very unusual for Cercospora. And the fact that flagellaris isolates infect Arabidopsis, I think, will be, a powerful tool to dissect what's going on. So the picture here taken at Silmar this year, for those of you that didn't make the meeting, the trees are almost all gone now. The phylogenetics meeting held out there every 2 years at Pacific Grove. This is pathology in action, unfortunately. And so the key contributors to the work that I was talking about today, so John Ridenauer is here. He's at Oregon State now as a postdoc with Michael Freitag. Wagner Fernandez is finishing up his PhD with Eva Stuckenburg at Max Planck. Alex Zacharone was my kind of bioinformatics technician. He's with Yanis Stergiopoulos, getting his PhD at UC Davis. And then Kona is my program tech who recently started her master's degree, who has done a lot of the work in the soybean Circospring, also led the effort on the Arabidopsis pathosystem. And a special thanks. So a few years ago, my lab is not really an Arabidopsis lab. We made a trip up, and Tess and Chow were very kind to patiently walk us through Arabidopsis, remedial Arabidopsis for beginners. And we were able to take that information and successfully translate it. And we've had so many people that have submitted soybean samples, symptomatic leaves, but from other things as well, that just couldn't list them all. But there may even be some on the call or in the room. So thanks to everyone. And this work's largely been funded by NSF and USD. So with that, I will take any questions you might have.
Okay, so it seems like, okay, so yeah, so for those of you connected, you can unmute yourself and ask your question. Perhaps we can start from here in the room. Any question?
I have a question.
Yeah.
So when you get your infections, there's sort of white lesions on Arabidopsis. Are those sporulating, or is it just the necrotic tissue?
Excuse me. I think I mentioned to some people earlier, I've got my second allergy season up here in Indiana, so just escaped it from Arkansas. But so those lesions will sporulate heavily, and basically perform Koch's postulates out of those to re-isolate and confirm and reinfect.
Nice.
So yeah, it's
good. Yeah, very, very nice work. Yeah. And your blocks, when I first saw your plot, I mean, sort of blocks and other stuff in there. The other obvious possible solution to me was that you had invasion by transposable elements. I assume you checked that, and that's not the issue.
Yeah, yeah, that's a great question, and it doesn't appear to be the case. Yeah, so like we don't see, in fact, in the Cercospora kikuchii and Sisbeckia genomes, they assemble extremely well with low coverage because there's so few repetitive elements.
Huh.
And we don't see an association there with.
Nice.
I have a question. A great talk. There's always one question that confuses me. Actually, I should say complex. So the inoculum is basically from either from seeds or from debris. So one would assume the infection occurred early in the season, but the symptoms only show up after the flowers. So I'm always kind of wondering what caused the pathogen change from bacterial phase to necrosis phase.
Yeah,
Can you speculate on that?
that's a really good question. And so in the case of CLB, maybe a little bit differently than frog eye leaf spot. So before his passing, Ray Schneider did some work where he looked at the length of that latent period. And the period of latency in Cercospora leaf blight appears to be a very, very long time, and usually long for Cercospora, many weeks in some cases. And one of the things that's really curious about the progression of CLB symptoms in the field is they appear to be highly synchronous at a field level. And so, you see a field that looks good, it starts entering late vegetative development, maybe early reproductive, and within a matter of days, it looks like, as severe as like an errant spray from a crop duster. And that level of synchronicity is really curious. And it would suggest, a relationship between plant development and the signaling between, perhaps as a soybean plant is entering reproductive development, it's dropping certain resistance, efforts that the fungus is able to capitalize on since it's there. The bronzing has been closely associated with the production of Sarcospring, which if Sarcospring were being produced during the latent period, you would expect to see that sort of bronzing much earlier. So, that's a long way of saying I don't know, but there's some things that kind of point to a developmental relationship with the plant as far as like the end of that latency period in the case of CLB. I find that explanation harder to believe for Cercospora saggina. If you scout soybean fields, the only place I've ever seen frog eye lesions on juvenile soybean plants is in Argentina, where they have had 20+ seasons of continual soybean on soybean with like minimal tillage. So the inoculum level is probably so high that you can see lesions on like very young plants. But in the Midwest and in the South, even in fields that have a really high history, so some of that being earlier season where the conditions are less favorable, so the disease is just kind of struggling along until temperatures and humidities rise. So this is a great epidemiological question for some of the people in the audience yeah, and I think that's, it's probably different for the two diseases, but yeah, there's a lot of gaps there.
And the cause plays a lesser role in frog eye than is conservative plant.
Yeah, and I think that's possibly also the case too, because with CLB, you tend to see that necrosis very much in the upper canopy, and frog eye begins lower in the canopy oftentimes and works its way up, which would suggest less of a reliance on the direct exposure to light.
Question here. So I'm thinking here about some potential practical implications of your work for the circadian rhythms in terms of inoculation. So for us, for pathologists. So for inoculations in the greenhouse or controlled environments, what considerations shall we have in terms of light, for example? Sometimes we don't have standards about light. What are your thoughts about it?
Yeah, so some of this will also, in my experience at least, will be dependent on the specific species. So in the case of Cercospora zeomatus, we see that light is less important than maintaining high humidity early on. So we even built some kind of simple chicken wire cages with plastic to maintain that humidity in growth chambers indoors. And the lighting is not, particularly, it's a fraction of the intensity of what we would see, in field conditions. In the case of frog eye, it's also fairly forgiving. That initial maybe 48 hours of humidity seems to be very, very important. But in the case of CLB, I suspect that part of the reason that, again, back to those different dynamics of the importance of Sarcospring, I suspect that some of the reason why we've never seen very good, and many groups have tried, and if anyone has, recently found a way to get excellent, consistent CLB inoculations in growth chambers of the greenhouse, I would love to know what that technique is. But so the most that we are able to generate with CLB is necrosis. So there's the spectrum of symptoms where you have this bronzing and kind of even purpling and leaf mottling. And necrosis we're able to induce, but we're never able to induce the classic sort of bronzing and purpling, that mottling. It's very, very difficult to induce. And so some of these light requirements may be, situationally dependent, but certainly if humidity isn't very high for the first 48 hours and the fungus isn't able to have, and I think we were talking at lunch that we've come to believe that kind of attractant cue that directs germ tubes or hyphae to stomata is probably a humidity gradient. And so if humidity is not controlled early on, that's almost guaranteed to make an inoculation fail in our hands. Maybe that's even more important than light.
Very good. Any last question?
But I have a burning question.
Oh, yeah, go
So
for
for
it.
the freak,
Yeah.
freak, if you knock out, it's knock out the indirect. If you overexpression to the artificial. What about like long day, short day mutation they're trying?
Yeah, so it would be,
If they have those like 19-hour clock.
yeah.
They want to have no SH, and they want to have become like 27 hours.
Yeah, we looked at like the DNA binding domain of CRP1 and looked at expression of frequency in that background, but we haven't looked at an altered period.
Yeah, freak, okay, also period.
Yeah, that's a really good question.
Very good. Well, let's thank Dr. Bluhm again.
Oh, I'm actually wondering who will get a haircut.
Yeah.
Okay.
For dinner tonight. So this one here is for alcohol.
Very nice.
Yeah, mitochondria. Typical, different mice.
I don't know if you guys have made
Mitochondria just rearrange all over.
You even don't know what's
Okay, well, I was just getting ready to send one, so.
I'll probably. I want to pick up that.
If you want to just go ahead and take care of it.
I will make a reservation. I will make a suggestion.
Okay, all right, thank you.
Yes, it's changed. Before we see the libraries.